Piet Stielesse is from Hasselt University, and I would like to tell you a little bit more about genetic susceptibility. It fits, of course, within the theme of this session, where you are studying the components that are important in development of disease. So both genetics and environment is, of course, important in your susceptibility to determine whether or not you will develop a disease. And I will go over some of the important principles, and I will use autoimmune disease as the disease that can be used to model all these concepts that we will be talking about. But in fact, everything that we will discuss in this lecture can also be applied to other complex diseases, so other diseases where both genes and environment are important in development of that disease. So this is basically the scheme for today. So I will start with a brief introduction on
I'm not doing exercises, but I'm just switching the light off.
Okay, that's better. I will give you a very brief introduction on autoimmune diseases, and then I will explain to you how we can identify the genes that are responsible for diseases. And I will do so first for very simple diseases, where only one gene is important in developing the disease. And then we'll try to explain to you how we can do that for diseases where we know that there are many genes important for the development of disease. And I will also end with some new approaches that are currently taken because of all the new information that comes out of the Human Genome Project. Okay, let's start with the autoimmune diseases. This is a long list of so-called autoimmune diseases. Probably you know that in autoimmune diseases we believe that the immune system is by mistake attacking our own tissues. That immune system should target, of course, foreign invaders, but in some cases the immune system by mistake starts to attack our own tissues, organs, and of course this may lead to pathology, to a specific disease. And in many cases these diseases are chronic. Here you see some examples of very rare but also more common autoimmune diseases. So, for instance, insulin-dependent diabetes or type 1 diabetes is very common, but we believe that this is also an autoimmune disease. Immune system attacks, for instance, the beta cells. Other diseases on the list, such as rheumatoid arthritis, are also very common diseases. 1 in 100 is the prevalence of rheumatoid arthritis. And you see, of course, that several components of the immune system can be involved in attacking a particular target organ, target tissue, and this will lead, of course, to the pathology that is specific for that disease. And in all of these diseases, we know that in the end, tissue will be damaged. Tissue damage will be, in fact, generated by different components of the immune system. Again, the same components of the immune system that we use to attack foreign invaders are here used to target our own tissues. And these can be antibodies. So in some cases, antibodies to self-antigens lead to tissue damage. In other cases, cytotoxic T cells can start to kill the own tissues. But also macrophages, they can also eat up our own tissues in some instances. And as you can see, all of these components of the immune system are in fact triggered by activation of T cells. As you know, the T cells are the main driver of the immune system, and in all cases, as you can see, T cells become activated in some way. T cells that are activated will induce one of these pathways that will lead to tissue damage. So T cells play a very important central role in the development of most of these diseases. However, we still don't know why these T cells are activated, how this is really happening on this part of the scheme, so why these T cells become activated in many cases still is not answered. In one of the diseases that we're interested in, which is multiple sclerosis, we know that T cells start a process that ultimately leads to tissue damage in the brain and the central nervous system. So in this case, we believe that in the blood compartment, T cells become activated. When T cells are activated, they can upregulate specific molecules which allow them to infiltrate into the central nervous system, for instance, into the brain. When these T cells are trafficking to the brain, they can be reactivated in the central nervous system, and this may then lead to a local inflammatory response that will lead to recruitment of other cells coming from the blood, but that will also lead to production of molecules, immune molecules, that will damage the myelin, which, as you know, surrounds the axons. And in MS, this is the primary hallmark of disease, that the myelin is literally eaten up by macrophages, or can be damaged by antibodies, or can be also complement can play a role. So you see many parts of the immune system that can lead to damage of the myelin, but also the myelin-producing oligodendrocytes and other cells that are involved. So basically, this is the scheme of events that probably takes place in a disease like MS, although I should stress that this is still not formally proven, so this is kind of hypothesis. And in many of these diseases, we still don't know exactly what is happening in the disease and why this is happening in a particular individual. In most of the autoimmune diseases, and systemic lupus erythematosus, or SLE, or lupus is another type of inflammatory disease which is presumed to be of autoimmune origin. In many of these diseases, we know that a particular genetic makeup is important. So there are some genes that increase your susceptibility to develop these diseases, but the genes are clearly not sufficient. In addition to those disease genes or susceptibility genes, you need other factors to develop such a disease. And in SLE, it's known that, for instance, components of the food can be important in developing the disease. Hormonal factors can be involved. Other environmental factors could be involved. And in many cases also infectious agents could be involved. Of course, infectious agents may also lead to activation of the immune system. In some cases, they may lead to activation of the immune system that leads to autoimmune pathology. But in general, we know that there's a complex phenomenon development of those diseases, and that both genes and environment is important in determining who will develop such a disease. And the genetics behind these diseases is still, in many cases, not so clear. And we believe that in many diseases there are several important genes that could play a role in developing the disease, and every gene on itself is probably not harmful, but if you have them in a combination, this may create an environment that leads to much faster activation of these autoimmune T cells. So when you carry those genes together with some environmental factors, they may drive the activation of T cells that leads to this pathology. For instance, it's possible that some of these genes could influence the expression of antigens in the thymus. And you may remember from the immunology that expression of antigens in thymus is important to develop tolerance to those antigens. So if the expression is reduced, the chance to develop or to become not tolerant to these antigens may increase, and in this way it could increase the susceptibility to activate T cells and start an autoimmune pathology. It's also possible, for instance, that there is less good inactivation of self-reactive T cells. So some of genes may play a role in this aspect. And when they all come together, they create an environment, like I said, that could stimulate development of, promote the development of autoimmune conditions. But again, the genes are not enough. There should also be some environmental triggers to develop such a disease. Let's come back to multiple sclerosis. Also this disease, we are still trying to figure out what is the role of genes, what is the role of environment. And here also immigration studies have been performed to try to understand the role of the environment, because MS is one of those diseases that is more frequent in the Western population. So you see the risk of developing MS is pretty high in the northern parts of America and also northern parts of Europe. In other regions, for instance in Africa, Asia, MS is much less seen. So apparently there is something in the air in Europe and America that gives you a higher chance of developing MS. And there have been some immigration studies, for instance, from people that have traveled, let's say, from India to the UK or back to find out what is really important in developing the susceptibility. And it was shown that if you travel from a young age, let's say from India with a low prevalence of MS to the UK with a high prevalence of MS, these children will develop MS at a lower frequency as if they were staying in India. But the children of these people, they will develop a higher chance of developing MS. So clearly there's also something in the environment that determines the susceptibility. One of the critical factors could be sunlight. This is data from a study in France where they have tried to correlate the level of sunlight with the prevalence of multiple sclerosis. And of course, as you know, in the south of France there is more sun than here in Belgium and the Netherlands, and you can see that the prevalence here is about 50 in 100,000, and it goes up to 100 in 100,000, and there appears to be a kind of correlation with sunlight. People have tried to understand this link, and there is some good evidence now that vitamin D could be an important factor. Vitamin D that may have an effect also on the immune system, and there is some evidence that lower levels of vitamin D, if you have lower levels of vitamin D, that you have a higher chance of developing multiple sclerosis. People are also trying to use this concept now to feed vitamin D as a potential therapeutic intervention. Two other factors have been demonstrated to be associated with MS development, two other environmental factors. One is infection, and there appears to be some correlation with Epstein-Barr virus, EBV, which is a very common virus, and many of us are infected with this virus and will not show any signs of this infection, but it also can lead to development of mononucleosis. And there have been some studies showing that if you have increased antibodies to EBV, that you have an increased risk of developing MS. Another known risk factor is cigarette smoking, so it appears that those who smoke have a higher chance of developing MS. These are data coming out of many different studies, but they all have two important issues that are not resolved. First is the mechanistic explanation. So why is it that if you smoke cigarette that there is a higher chance of developing MS? There are quite a lot of theories behind it, but there's no proof what is really important there. And sometimes it's also hard to figure out whether some of the things are really causal, whether they are involved in developing the disease or merely are associated with the disease. So that's very hard to figure out. But like I said in the introduction, in all these cases genes are important. And the question, of course, is how can you study the importance of genetic factors in such diseases? So people look to familiar clustering. Of course, when you find families with high prevalence of disease in a pedigree, then it gives you already indication that it runs in families, so there may be a genetic component. Although you always have to be careful because families usually also grow up in the same environment, so you never know, are it the genes that are important here, or is it the environment that is also shared between people and the family? So that's why in some cases people have also looked to twins, especially monozygotic twins, because they share most of the genetic makeup, and then you can see whether if one of those twins will develop a particular disease, will the other twin also develop that disease with the same genetic makeup. And people also look to see the frequency of a disease in siblings versus general population. So this slide tries to explain that indeed for some diseases we know that the genetic makeup is important, so this will also be translated in a higher concordance rate in monozygotic twins. Maybe I can already show you this slide here. For some diseases where you see the concordance in monozygotic versus dizygotic twins, of course they will share most of the genes, or all the genes, most of I should say, and these will share some diseases, some genes, and if you then compare the frequency that in some cases goes up to 100%, indicating that genetic makeup is really important in developing disease. If you have the same genes, then you will develop that disease. In other cases with the same genes, for instance in psoriasis, still if you have the same genes, it will not mean that you will also develop that disease, so your chance of developing that disease is 60%, which is pretty much higher than for dizygotic twins, so indicating genes are important, but they don't tell you everything. What about MS? So the overall risk of developing MS in the population is about 1 in 1,000. For adoptive siblings, this is the same, indicating here again that if you don't have that the genes of that family, that you have a risk in the general population. But if you have a cousin in the family that has MS, then the chance of developing MS will slightly increase up to 7 in 1,000. If you have a brother or sister that has MS, your chance of developing MS will also slightly increase up to 35,000. But if you have a monozygotic twin with MS, then you will have a chance of developing MS about 1 in 3. Again, illustrating the point, even with the same genes, the chance of developing MS is not 100%. It's much higher than in general population or even with siblings, so indicating genes are important, but they don't tell you the whole story. Okay, so if we know that genes are important, the next question is how can we identify those genes and what can we do with that type of information? And to make the story not too complex, we will start with simple diseases. So we do have some rare autoimmune diseases where only one gene is important in the development of that disease. And let's start with these examples. Of course, you could ask the question, why do you study such diseases if they're so rare? Well, we know by some examples that if you can find the genes that are important in these rare monogenic diseases, that they can learn you something about the more common complex diseases. So these genes will provide you information to give some more understanding about the disease mechanism, and it can be also be useful in the more common disease. So how do we do that? How do we find those genes? Well, basically, we follow here the positional cloning strategy. So the idea is that you try to first pinpoint the position of the gene on the genome. So you try to identify the chromosome or the chromosomal region where such a gene is located. And once you have this location, then you can use various technologies to identify the genes in that particular region, sequence the genes, and find the mutations that are important. So how do we do that? What is positional cloning? So like I said, the first thing that you have to do is try to identify the position of a gene on a chromosome. And we do that actually by using markers which are positioned on the different chromosomes in our genome. And if you know the position of markers on a chromosome, then what you can do is try to identify the particular marker on a particular chromosome or chromosomal region that is close to your disease gene. So if you know that the Z gene is, for instance, close to this marker, then you know the position. If you know that's another close to another marker, then you will also know the position of that gene. So how do you do that? How do we measure position or distance in genetics? We can do that by measuring recombinations. You know, in every meiosis there can be a recombination, and this can be seen, of course, if you study pedigrees. So recombination is studied in families. So here you see a simple example. Let's say this is the disease gene, and this disease gene is close to this marker on this chromosome. And as you can see here, in this case, this sibling son, the son or daughter from this father has also received the same chromosome. So he or she has the disease gene, but also this particular marker, which is close to that disease gene. Here you see another example where this individual has received this chromosome with the disease gene and also the particular marker. So they appear to be close to each other. If you know the position of that marker, if you would know that it's, let's say, on chromosome 21, on this particular region of chromosome 21, then you know where your gene is located, because they apparently are close to each other. They are inherited together in a pedigree. On the other hand, you also see that there is one example here where this chromosome probably came from the father, and where you see the marker, but you don't see the gene. So indicating that here a crossing over has appeared, indicating that this part of the chromosome was connected with this part of the chromosome. And in this case, so this chromosome has been reshuffled, and the marker is there, but not the gene. So this indicates that they're somewhere close to each other, but they're not very close, because there is still a possibility for recombination. And as you know, when two genes are close to each other, the chance for recombination is much lower than when genes are far away from each other. So this is a way to measure distance by looking at recombination. It gives you a sign of the distance between two genes. And you know, in genetics we can calculate that also as centimorph, and I will come back to that in a minute. So this is the principle. Look for a marker that is inherited together with your gene in a pedigree. Okay? Now the problem, of course, is that you should be able to measure recombinations in such a pedigree. And you can only, I mean, like these two chromosomes, if you see them like this, you can never find out whether there is a combination here because these two chromosomes look the same. So what we try to do in genetics is make a distinguishment between the two chromosomes. If we can find differences between the two chromosomes, then we can look to recombinations. So basically what we try to do, let's say this is the disease gene in green here, and what we try to do in this pedigree is make a difference between the blue chromosome and the red chromosome. If we have ways to discriminate these two chromosomes, then we can follow the fate of those chromosomes in a pedigree. And you can see here that all individuals that have received this part here in red, they also, of course, have received that particular disease gene. You see it here, you see it there, you see it there. And you see that there's a part of this chromosome that is in this chromosome, this reshuffled chromosome, but there's also a blue part missing the green part. So if you can discriminate the two chromosomes, of course we use DNA markers to do so, then you can measure recombinations, and you can look for, regions where your gene is located. Maybe I'll come back to that in a minute. But to do so, we need those markers which are positioned on the chromosomes. You're very lucky because these days you have all these maps available on the internet. You can find for every chromosome a genetic map on the internet with different markers, as you can read here, that are positioned on a chromosome. And you can see that those markers are positioned also with the genetic distance between them. And this genetic distance is listed on the genetic linkage map. And genetic distance is measured in centimorgans, which is percent recombination. Okay, so keep in mind we need those maps with all these markers to identify the position of our gene. Like I said, you have today all these detailed maps of all the chromosomes with many of these markers, with the genetic map of each chromosome, and also the physical map of the chromosome. Genetic maps and physical maps, they may look a little bit different. Of course, the order of the markers is the same, but in some areas of chromosomes you have a higher chance for recombination than in other parts of the chromosome, and this may be seen as a difference between genetic distances and physical distances. But I will not go in detail to explain you this. The principles: we have a map, markers, and now we try to identify the gene. So first we start with a pedigree, and you have to figure out what is the inheritance pattern in such a pedigree. Is it recessive? Is it autosomal? This is important to find out whether you need one gene or two genes to become sick, to develop a disease. Okay? You can see that by just looking to pedigrees, what is really the inheritance pattern. And then what you do is you take DNA of all these individuals, and you start analyzing this DNA by looking to all these different DNA markers positioned on every chromosome. And what you basically do is you start, let's say, on the tip of chromosome 1, you go over chromosome 1, then you use markers from chromosome 2, chromosome 3, and so on. You scan the whole genome to look for a marker that segregates, that is inherited together with your gene in a pedigree. If it goes together with your gene, you have few recombinations, and this can be analyzed with a computer program, and this computer program will show you where the gene is positioned. And what we basically try to identify is two markers on each. If this is the gene, then we need two markers on each side of the gene, so two flanking markers, because then we know exactly where the position of that gene is located. So this is a typical outcome of such a genetic analysis running out of the computer. And it shows you, doesn't matter what kind of disease, this is just an example. It shows you this plot with, as you can see here, distance in centimorgans, so genetic distance. And what it shows here is lot scores. I think on the next slide you will see the definition of lot scores. It's a logarithmic score which gives you a chance, probability, that there is linkage versus no linkage. And what you should know is that once you have a lot score of higher than 3, which means that you have 1,000 higher chance of linkage than no linkage, then this is considered to be as a good evidence that there is linkage in this area. And here you see in this plot that actually this marker probably have a lot score of around 3, indicating that you see the chance that your gene is located here is pretty high. This is known from this genetic analysis. We know this marker is on a particular site of the chromosome, so we know now that the gene is located close to this marker. What is it? D17. 17 stands for chromosome 17, and then they all got a specific marker or number. S, I can read it, but this is the marker where our gene is close to. What you also see from this slide is that these two markers are flanking the region. So what you basically can say now is that your gene is within this region. Flanking this marker, probably close to this one, but you don't know if it's on the left-hand side or the right-hand side. It's within this region. And once you have this information, then you can use a battery of technologies to identify, clone out a gene. So if you have a flanking marker on this side and on that side, you can use other technologies. We don't go in details, but you can use chromosome walking, jumping. Today you have all these genes, of course, somewhere in the library. You can really clone out all the region, you can sequence that region, and you can look for the genes. And you know, in the DNA sequence, it's not so easy to find the genes, but there are several ways to do that. You can use computer programs to do so. You can use cDNA libraries, Northern plots. These are all methods to identify the exons or the genes within such a region. And once you have identified, if you're lucky, there is only one gene. If you're in problems, then you can have, let's say, up to 10 genes, and it becomes more difficult. But let's say you have only one gene, then you can look for the gene sequence and identify mutations just by comparing patients and controls. So this is the way you can go to identify these genes. What can you do with that information? So once you have the gene, of course, you can do all the diagnostic studies. You can identify individuals that carry such a disease gene or that carry such a disease mutation, and you can do that before the disease will appear. So before symptoms of disease will appear, you can do that prenatal, or you can even use other ways to diagnose for the presence of this disease. And of course, what people also try to do then is to find out what is the function of disease, of these genes. Because in this approach, you have not really looked at the function of disease. You just looked at the position of the gene and then picked up the gene. And now you're interested to find out what is really the role of those genes. And then, of course, you can use knockout models or other technologies to find out what is the role of this disease, what is the role of the gene, and what is also the role of that gene in the disease. What's next? Of course, what people hope to do is to find therapies that can target that disease. So this approach, positional cloning, has been used in the 80s and 90s to identify all genes that are important in monogenic diseases, in these Mendelian inherited diseases. So today we know all these genes. This work has been done. I think that most of those studies were published a long time ago, using this approach to identify this, such genes. And of course, this was the simple part. Today, I was hesitating whether I should explain to you positional cloning, because you see it's an old way of finding genes. And of course, you are coming now into the area of whole genome sequencing. And I'm not sure if you know Nick Volker. Does anyone know Nick Volker? He's probably the first boy who has been treated by identifying his mutation without knowing the disease, just by sequencing his genome. So it's a very nice story. You can read it all on the internet. And they say, okay, he was lucky and unlucky. This boy, he was suffering from a very aggressive bowel disease, and his parents and also medical doctors didn't know what was happening here. They were thinking of really taking out his complete bowel. There was no indication what was happening. He was kind of, well, it was very aggressive, so people were thinking that he probably could have died from this disease. And he was lucky because his parents, they really wanted to find out what was happening, and they went to a genetics center. And so what they did was they sequenced his whole genome. With this analysis, they found out that he has a mutation, a particular gene on X chromosome. You can read it on the slide. It turned out that actually this gene was important, probably in provoking an autoimmune disease against his own bowel. So they decided that they should replace his immune system. He got a cord blood transplantation, and it turns out now that he is safe. He has no problems anymore with his disease. So as you can see, he can eat everything. And this is considered to be the first boy that really is treated by finding the gene or the mutation that is causing his disease without any knowledge about his disease. So that's why I told you I was hesitating to tell you all the story about positional cloning, because these days we have the whole genome sequencing technologies, and they can of course also be used to identify particular genes in a particular disease. And you will hear more about this of course in years and months to come. There's also a nice story, because I think the journalist who wrote the story got an important prize in the US, and I'm sure that there will be a film in 1 or 2 years from this story. So let's go back to autoimmune diseases. What did this work show us about single gene disorders? Like I said, these are rare diseases. So usually have one gene, of course, that leads to development of that disease. And I will give you 2 examples of such rare diseases where they also have identified the genes, and those genes will show us something about the mechanism of autoimmune disorders. What is autoimmune polyendocrine syndrome, APS? And as you can read, it's an autoimmune disease. And people have identified a mutation that was important in development of this disease, and it was a mutation in what's called the autoimmune regulator. And this appeared to be a protein that was responsible for the thymic expression of antigens. Again, expression of antigens in the thymus is important because it will induce tolerance to your T cells. Your T cells that will see antigens in the thymus, they will get depleted, so they will become tolerant. If there is a gene that reduces the expression of those antigens in the thymus, of course this will increase the susceptibility to develop autoimmune conditions. And if this protein will do so for a wide array of autoantigens, then of course you may develop reactivity to different antigens, different organs, different tissues, and usually it leads to a widespread pathology. As you can see here, organs, skins, and other tissues can be attacked in this disease. So in this case, identifying the gene gave us a protein that appeared to be very important in thymic expression, and is of course also important to understand the role of these proteins in the immune system. Second example is a disease which is called IPEX. I'm not going to read the whole story here, but you can see that there is a lot of immune pathology in this disease. Again, it's a very rare disease. And people have found that there is a mutation in FOXP3. Now you may have heard of FOXP3 before, because FOXP3 is a very important transcription factor that is expressed in T cells which have a regulatory suppressive capacity. So T cells that regulate the immune system, they express FOXP3. And FOXP3 appears to be a very important gene for making T cells suppressive or regulatory. There's also a mouse model where the same gene, if you knock out, then you also see a very systemic autoimmune pathology. And again, there are no Tregs in this disease. So this brings everything together. Human disease with a mutation in FOXP3. FOXP3 is important in T cell regulation. If you knock out FOXP3, you get indeed an autoimmune pathology with no regulatory T cells. Probably this also is important, of course, in the human situation where mutations in FOXP3 may lead to less regulatory T cells, and if you have a problem with regulatory T cells, this may help the development of autoimmune conditions, because the regulatory T cells are no longer there to suppress the aggressive T cells, and then the aggressive T cells can, of course, induce disease. We've looked at this molecule also in relation to multiple sclerosis. And our group some years ago already demonstrated, also together with others, that there is a problem in T cell regulation in multiple sclerosis. So we could demonstrate that if you look to the regulatory T cells in MS, that they appear to be dysfunctional in a large group of MS patients. You can see that also from this slide, showing that the suppressive activity or the regulatory activity of T cells is significantly reduced in healthy control in MS patients as compared to healthy controls. And we could also demonstrate that if you look to FOXP3, that these T cells from MS patients have a reduced expression of FOXP3. So FOXP3, again, important marker for regulatory T cells. If it's not working as it should, it may lead to a situation where autoimmune pathology can appear. So this is also a gene coming out of this rare diseases that plays a role in the more common diseases. So as a conclusion, we can say that even the study of the rare human autoimmune diseases and specific mouse models have not focused on that, but they provide information that helps us to understand development of autoimmune conditions, autoimmune diseases, and this may also help us to identify genes that are important in various autoimmune diseases such as type 1 diabetes, rheumatoid arthritis, multiple sclerosis, and others. Okay. So if this is a simple situation, let's go then for the more difficult situation, and these are the more complex autoimmune conditions. I was wondering, I was planning to take a 10-minute break. Is it a good thing to do this right now, or can you continue for some time? Still possible to go? Okay, I will go ahead a little bit more and then break for 10 minutes later. Good. So for common diseases, and it stands here common autoimmune diseases, but it's also true for many heart diseases or cardiovascular diseases or some cancers. These are complex diseases because we know that many or different genes are important. But in many cases we have little information to really understand what we're looking for. Are we looking for a limited set of genes that is important in all patients? Or is it a wide array of genes where some genes are important in this patient and other genes are important in another patient? This is the type of information that we don't have at the moment when we are looking or starting to look for the genes that are important in these diseases. We also don't know the interaction with the environment, so it's possible that in some cases you need to add up some genetic susceptibility factors to increase your chances, but still the environment is important to really determine whether you will develop that disease. So this, of course, makes life extremely complicated. And the way to identify these genes is sometimes a little bit different than what I said before from the simple one gene disorders. So in the one gene disorders, we studied families. Yeah? What we basically did was looking for linkage. Because if we have a marker on a chromosome that's close to your gene, we could call that linkage, they're linked. And this linkage analysis or positional cloning is typically something we do for these diseases where only one gene is important. You can also do that for more complex diseases, but the main problem is, of course, that in linkage analysis you need families. And in many of these diseases you don't have large pedigrees with many individuals suffering from that disease. So the difficulty is finding the pedigrees. So that's why people have also looked at a different way to identify genetic proximity, and this is by doing association studies. And I will try to explain the difference between association and linkage. In association studies, we look for linkage disequilibrium. Now, what is linkage disequilibrium? I think I can best explain this with this example. Let's say this is these are two parts. These are two same chromosomes. Let's say chromosome 2, and there are two regions which are where you have two alleles. It can be a gene or a region of this chromosome. Let's say here at this position you can have this big A or small a, and at this position you can have big D or small b. So they are on one chromosome, and these are also on one chromosome. So these, of course, are called alleles. And then you can measure the frequency of each allele. Let's say that the big A has a frequency of 30%, the other one 70%. This one has a frequency of 45, the other one 55. So you know the frequency of the alleles in your population. Then what you can do is calculate the haplotype frequency. What is a haplotype? That's a combination of alleles on one chromosome. So one haplotype is big A, big B. Another haplotype is small a, small b. But of course you could also have the haplotype big A, small b, and small a, big b. These are the four possible combinations, the four possible haplotypes that are available. And then you can take a group of individuals. Let's say you will screen 500 random selected individuals, and you will genotype them. So you will determine whether they have this allele or that allele on one side of the chromosome and the other alleles on the other side of the chromosome. So you will genotype all the individuals in your population. And based on the genotypes, you can calculate what is the frequency of each haplotype. For instance, let's say this would be the the genotype of an individual, homozygous for this big A, homozygous for this big B, then you know, of course, that he will carry one haplotype AB and one haplotype just big A, big B, and one haplotype big A, big B, so 2 times this haplotype. If another individual would have this allele combination, then you will know, of course, there should be one haplotype like this and one haplotype like this. So this one will have one time this haplotype, but also the haplotype small a, big B. Just as an example, you do that for all the individuals, and in this way you can calculate the observed haplotype frequencies. You can calculate for each of these haplotypes what you see in the population. And then you will calculate what you would expect. You can also calculate by just using the allele frequency. So this combination would be then this frequency times this frequency multiplied by this frequency. And this will lead you to these expected frequencies. And then you just compare to that. You compare the expected and the observed. Of course, you use sophisticated statistics to see if there is a really significant difference. I've given you an example where you can see it, of course, just by the eye. Here you see that the frequency of this haplotype would be around 16%, but it appears to be 55%. Way higher than what expected. And so on the others, here for instance, you would expect 38, but it's only 7%. Way lower. So it appears that these 2 genetic regions, they are segregated in the population at random as you would expect. If they would segregate at random, we'd call that a linkage equilibrium. So they're not in equilibrium, they're in disequilibrium. That's why we call it linkage disequilibrium. And very simple, if 2 regions are in genetic linkage disequilibrium, they're close to each other. Because it usually indicates that they're in the same genomic region. So association measured by linkage disequilibrium is another way at looking at genetic distances. If 2 regions are in linkage disequilibrium, they're close to each other. Okay? I hope this is more or less clear. So again, to summarize, linkage disequilibrium, 2 alleles of 2 genetic variants occur together on an individual chromosome more often than expected by chance alone. For instance, they are more often expected here than what you would expect by chance alone. These variants are just associated, and usually this linkage disequilibrium also means that these markers are very close to each other. We're talking here about distances from 5 to 20,000 bases. While in linkage analysis, you're talking about centimorgans, which can be millions of bases. So once you have here found a marker which is close, then it's really close. So this is a measure of close distance, and I showed you how you can do that. And linkage, just to give you the difference, so linkage disequilibrium, remember this is association. Linkage, this is done in pedigrees. Linkage, again, linkage disequilibrium, we don't need pedigrees. We can do that in the general population. But here we need pedigrees, and we're looking for markers which don't recombinate much, and that also means that they're close to each other. Another way of looking at this, these, you can see with the colors that these alleles or markers are in linkage disequilibrium. If you see here, there's the blue and the red one, the green and the gray. If you look at the population, you will see always the blue and the red one coming together, and the green and the gray one. Okay? You don't see much exceptions to that. So this indicates that there is linkage disequilibrium. They come together. Here you have 2 markers which are in linkage, meaning that they're close to each other, but they're not in linkage disequilibrium. So if you look in the general population, despite the fact that they're close to each other, you still see all combinations possible. This combination with the white one, but you will also see this combination with that one, or this combination with black and white. You see all possible combinations. Just by looking to this population here, you will not be able to say that these markers are close to each other. Here you can say that because you know that they tend to stick together, alleles. Okay? So, what do you need to do to find out that these markers are linked? Study pedigrees. So if you go to pedigrees with these markers, then you will see in the pedigree that there is not much recombination between those 2 chromosomes here. You see there's still one recombination here, black and white, but in general they don't recombinate often. Just to show you that linkage is done in pedigrees, association is done in populations. Okay, so far we've talked about recombinations, association, alleles, but I've not explained to you what are really the markers that we use in genetic studies. You know that, of course, that we need polymorphic markers to discriminate the green chromosome from the red chromosome. In the lab we cannot follow some of these chromosomes, so what we do is we use polymorphic markers, so regions in which you have different combinations. And you know that the polymorphic markers that we use in genetics, for instance, are RFLPs, sometimes also called RIFLIPs, restriction fragment length polymorphisms. This is a very old name, but people are still using solid blot technologies, for instance. Today, most of the polymorphisms that are used are either SNPs, SNPs, single nucleotide polymorphisms, where a particular region or a particular chromosome, someone will have, let's say, an A in the sequence, and other people may have a C, for instance. So some will have the allele A, other will have the allele C. This is a typical SNP. One nucleotide difference. The other type of polymorphisms that is frequently used are called short tandem repeats. This is the sequence. It can be up to, let's say, only 2 nucleotides, or 3 or 4, but it's a sequence that is tandem repeated on a chromosome, meaning that you have the sequence, let's say, CA, 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 after each other. And what is here the polymorphism? What is the difference between alleles of different chromosomes? Well, if you look to a particular repeat on a particular chromosomal location, there are some people may have, let's say, A times CA. Other people will have on the same spot, may have 12 times CA. Other people may have 14 times CA. So what you see is the difference in the number of repeats in such a region. And this is also called the variable number of tandem repeats. Another way to look at it is, another name is the microsatellites. But these are also polymorphic regions. This is an example with a SDS page amplification of these STRs. So here you can read these alleles are different individuals. And here they have amplified 3 different of these STRs, or these regions where you have this amplified sequence. And as you can see, for every individual you have 2 bands. So there's also a shadow band, but basically you have 2 bands. And these 2 bands, they correspond with a particular length, typical number of repeats of this particular repeat. So here, for instance, you have allele 11, here you see you have allele 15, and so on. You see that there is wide variety among these individuals, which is nice, because what you try to use these polymorphisms for is to find differences between chromosomes. And of course, if you have a lot of possibilities, the chances to find differences between 2 chromosomes is way higher. So these STRs are very useful because you have many different alleles. That's different for the SNPs. The SNPs, usually you have only 2 alleles. For instance, some people have a C, other people have an A at that position, or a T, G. You see that usually you only have 2 possibilities. You may have more, of course, but usually you find only 2 possibilities. And today we have this maps of the whole genome with all the SNPs in the different locations. And you also have the technologies to look at the SNPs. We can do that with DNA chips, as you probably know. You can analyze the genotype of many different SNPs of different individuals. And again, you have these beautiful maps of different chromosomes, which are extended every year, and where you can see on the map, the genetic map again of this chromosome, the physical map. So this is the true length in basis or megabasis. This is the length of the chromosome in recombination frequency in centimorgan. And then you have all the markers in between to show the position both on the genetic and on the physical map. And here you see the number of SNPs that you have in every different chromosomal location. There's a huge number of SNPs available today for genetic studies on these chromosomes. So if you have to choose between using SNPs or STRs for your genetic studies, you have to consider that SNPs usually only have 2 alleles, which may not offer you always a lot of information, because there's always a chance that people, of course, have the same allele, if there's only 2 combinations possible, while the STRs, then you have many different alleles. So the chance that you can discriminate between chromosomes is much higher when you use STRs than when you use SNPs. On the other hand, SNPs are more frequent in the genome. So you have many more SNPs on each chromosomal location as compared to STRs. And with the SNPs, you also have automated analysis. So you can use DNA chips to analyze them. In some cases, these SNPs, these are just single nucleotide changes or differences, they can be located in an important region of a gene. They can, for instance, be located in the promoter element or regulatory element or even an axon. And in that case, of course, such a SNP may also have a functional difference. In many cases, we look to DNA polymorphisms that have no influence on the function of the individual, so they have no functional consequence, but some SNPs are located in axons and may have a functional consequence. Of course, these are also interesting to use in these type of genetic studies. I think it's better maybe to use this 10-minute break now. Is that okay? Yeah? So I see it's, let's say 10 minutes, come back, 5 months.